Hello children, today there was an interesting topic, that is photosynthesis. Actually, regarding this topic, you might have come across like structure of the chloroplast and the types of the pigments. And also we are all very much well known that the plants are synthesizing the food material. We are getting a lot of food material from the plants, isn't it? Then one amazing thing is how the synthesis takes place in plants. It's a really amazing thing. That will go for what? Let's move. Okay. Before going to the process of photosynthesis, let us know about quantazome. What is quantazome? The quantazome is nothing but the collection of pigment molecules which are help in photosynthesis. The pigments which are help in those pigments which are help in the photosynthesis. And this quantazome is also called as unit of photosystem. This unit of photosystem has a single chlorophyll A pigment. A single chlorophyll A pigment, and which is surrounded by many accessory pigments. These pigments may be either xanthophyll, carotenoid and chlorophyll, other chlorophyll pigments like chlorophyll B, chlorophyll C, chlorophyll E, D, whatever it may be. But the chlorophyll A is a single one. The unit has only a single chlorophyll A pigment. When light falls on these pigments is also called as accessory pigments, which are surrounded by chlorophyll A. This is a single one where actually the reaction starts. That's why this chlorophyll A is called as reaction center. This is called as reaction center. Okay. When the sunlight fall on these pigments, different kinds of pigments like xanthophyll, carotenoid, chlorophyll C, B or D, whatever they may be, they receive different types of the pigments, receive the different types of the wavelengths. So the light which is fall on these pigments is in the form of photons. This is in the form of photons. And when these photons fall on the pigments, these photons energy is transformed from one pigment to the another pigment. That means, suppose the photons fall on this first pigment, then it is transported to next, and here to here next, and the energy passes from one pigment to the another. Like this, the photons moves, and ultimately the energy is reached the chlorophyll A pigment. Then what happens? The energy which ultimately comes to the chlorophyll A pigment get excited, because so much of energy is present in the chlorophyll A pigment. So which result the energy get excited. Then what happens? When the energy get excited, a pair of molecules are received by an acceptor. This is called electron acceptor. Why? Because the electrons which are excited from the chlorophyll A are received by an acceptor. These electrons, a pair of electrons, are received by electron acceptor. So, this entire process is called as quantazome, or the unit of photosynthesis, the synthesis. And then what is, suppose if the chlorophyll A is an isolated condition, then what happens? It is in an isolation condition, then what happens? Here, that means isolated condition means this we are doing experimentally, we are doing here, but it is taking place in the cell. Here it is outside of the cell. That means experimentally when we are doing this process, the electrons which excited are not received by any electron acceptor. Then what happens? Then these electrons are having a tendency to come back to the same. That means higher to lower. This chlorophyll is having lower energy level. Why? Because already the pair of electrons are get excited and they move away. So this is having, now it is in a lower concentration level. So these electrons are having a tendency of moving again to the chlorophyll A. Then what happens? During the movement of these electrons, a light is emitted. Some amount of energy gets emitted as a different color. That process is called as, that means the wavelength emitted. Whatever the color may be emitted. This process is called as fluorescence. Fluorescence. That means this is only possible when this process takes place outside of the cell. That means experimentally when we do this one. Otherwise, in case of when the process is taking place, there is an acceptor which receives the electrons, and this is a quantazome. And then next, okay, now we understood about the structure of the quantazome. Now, when the light falls on the pigments or the chlorophyll A, how this chlorophyll behaves? There is a two system, that is PS, photosystem 1 and photosystem 2. These are the two actually main systems. Whatever the system may be, system is having a chlorophyll A that we have already discussed, chlorophyll A, and then this chlorophyll A is surrounded, this single chlorophyll A is surrounded by many pigments that we have already discussed in the structure of the quantazome. Okay. And what happens here, when this light, this is the reaction center chlorophyll, and these are accessory pigments. And together these reaction center and accessory pigments is called as antenna complex. These two are called as, I'll write it here. Sorry. These together called as antenna complex. Antenna complex. Then we already discussed this. When sunlight falls on the pigments, these pigments absorbs the photons, and these are pass on to the next, and ultimately this is the photons is passed to the chlorophyll A. The chlorophyll A, here the important point you have to remember is The chlorophyll A absorbs the wavelength about 700 nanometers. This is the important thing you have to remember. In the photosystem 1, when the sunlight fall on the chlorophyll A and this absorbed at 700 nanometers. When the chlorophyll is having a complete energy, this chlorophyll A would get excited, which results, that means there is a pair of electrons would be get excited. Here these electrons are received by an electron acceptor, because this is the first electron acceptor which receives the energy. That's why this is called as primary electron acceptor. And the entire system is called as photosystem 1. That means the main thing here it is, in the photosystem 1, this chlorophyll A absorbs the photons at 700 nanometers. And in the same manner, photosystem 2 is also having chlorophyll A, single chlorophyll A, and these are surrounded by many accessory pigments. And in the same way, whatever the systems may be, when the light falls on this, the photons, or when the sunlight falls on, these photons are passes from one to the another, and finally these photons will be passed to the chlorophyll A. And here the difference between these two is, in the photosystem 2, when the photons are received by chlorophyll A, this chlorophyll A absorbs the photons at 680 nanometers. So the same thing is happening here also. When the chlorophyll A is receives the complete energy, it would get excited. Then here also it is received by an electron, that is primary electron acceptor. A pair of electrons would get excited from the chlorophyll A, and then these electrons are received by the primary electron acceptor. Whatever these two systems may be, the same structures you are finding here, but the main difference is, here in the PS1, these photons absorbed by chlorophyll is 700 nanometers, and in the PS2, the photons received by the chlorophyll A is at 680 nanometers. And one more thing is, this photosystem 1, simply we can write as photosystem P700, and PS2 we write like P680. That means photosystem 700 and photosystem 680. Okay, and here in the either it is a PS1 or a PS2, the chlorophyll A is a one and only the reaction center. In the PS1, the protons are absorbed by 700 nanometers. That means the reaction center, chlorophyll A absorbs the photons at 700 nanometers. So this is reaction center. Here also the chlorophyll A is a reaction center. But the one difference is, it receives the photons at 680 nanometers. So this is about the photosystem 1 and photosystem 2. And next class will go for the how the reactions will takes place, how the synthesis takes place. Thank you.